Hello everyone, welcome to PhDMonk, the show where we try to talk about stories, struggles, and curiosities of people involved in research. I'm your host Ashutosh, and today we have someone who's making a big waves in the world of science. My guest is none other than Professor Helen Walden. She's a professor at University of Glasgow. She's a structural biologist. Her work helps us understand how cells control proteins, basically ubiquitination, something that's super important for figuring out diseases like cancer and Parkinson. In today's episode, we'll talk about how she got into science, what drives her into research, and what life as a scientist really looks like, from her lab experiences to mentoring young scientists. So please welcome our professor, Professor Helen Walden. Uh, Welcome, ma'am.
Thank you very much, Ashutosh. Nice to meet you.
Nice to meet you. So, ma'am, I will start with the a basic, very basic fundamental questions. Like every one of us is get interested in science from a moment or a life experience. Do you, have anything when you started science, or just you were always interested in doing science?
Yeah, so I think there wasn't a eureka moment for me when I realized that I really wanted to do science, not as a child anyway. But what I would say is that I was always very analytical. I always wanted to understand how things work, how people work. I just always had this curiosity about how things work. And so when at school, I was pretty good at languages, pretty good at science, and had to decide what I wanted to do next. I thought I found science a bit more challenging than I found some of my other subjects. And I thought it would be more fun to do something challenging than something I found very easy.
So, yeah, like once you completed your school education, so you went on to University of Bath. So, is this a particular like you started thinking that you will be doing science from there, or uh, how is the experience there?
Yeah, so I was the first member of my family to go to university, so I didn't know what university really was. I just knew that I was going to go, and I thought biochemistry sounded very interesting. So I applied for a number of biochemistry courses at different universities around the country. And actually, I didn't make the entrance requirements for um, Bath, which was my top choice. And I remember being very panicky that I wasn't going to get in. And I phoned the admissions system, and they said, nope, it's absolutely fine. You can come. So at that time, Bath was a fairly young university, but it had a strong reputation in sciences, particularly applied sciences like engineering. And so, yeah, I went there and I fell in love with biochemistry almost from the first day.
So, what inspired you in your graduation? Like, which part of the biochemistry do you like the most there?
So, in terms of subject area, I very quickly was very interested in structural sciences. Um, I think the physicality of them, the kind of what's the word? The sort of realness of them really appealed to me. And one of the things that was really great about the Bath biochemistry course was that it had two sets of 6 months um, research, uh, research projects. And so the way the course was structured was that in the second year, you got to go somewhere for 6 months to do research. And then in the third year, you got to go somewhere for 6 months to do research. And then in the fourth and final year, you did a final year project. And when I didn't do very well in my first year, and so I was left with not very interesting possibilities. So I ended up working for a company that manufactured diagnostic kits. Um, and my job was to test a particular component of that kit for cross-reactivity. So I did the same experiment every single day. And I thought, wow, this is not what I want to spend my life doing. Um, and so I knuckled down and I did much better the following year. And that meant that I had more choice of where I went for my placement. And I ended up at the Weizmann Institute in Israel. Um, and I had a proper autonomous research project to do there. Actually looking to see how the heat shock response, how molten globules would induce the heat shock response in xenopozoocytes. So a long way from what I actually do now. But I remember just being absolutely fascinated, couldn't wait to go in the lab and see what happened to my eggs and see what color they had turned. And I just really loved it. And I remember coming back from that experience. My dad picked me up from the airport, and even before I said hello, I said, I want to do research. I know now that I want to do research. So that was really transformative for me.
Yeah, because a lot of my friends work in industry as well. So they always say that it's a monotonous life. You do every single thing every day, and even if the highly passionate and interested person might be able to at a certain period of time might lose some interest in what they wanted to do and in what they are doing.
Yeah, I think that's true. I mean, academic science can be incredibly monotonous as well, doing the same experiments. But I think with any setting, as long as you can keep the goal in mind. And I guess maybe I didn't want my goal to be making a good kit for a hospital, right? I didn't want to be manufacturing something. I wanted to be learning something, right?
So then later on, you went on doing your PhD. So how what were you working on in your PhD, and how your previous experience as working in different projects in your bachelor's also helped you in your PhD tenure, and what did you take give big takeaways from your PhD life?
Yeah. So I was really fortunate with my PhD because I had been working in my final year project in a lab that specialized in understanding adaptation to extreme environments, usually metabolic enzyme adaptation. And although the lab I was in weren't structural biologists themselves, they collaborated with a structural biologist called Gary Taylor. Um, and while I was doing this project, I think I was just investigating the effect of salt on the stability of a particular enzyme. Um, I realized that I think I realized that the answer would lie in the biochemistry, the actual chemistry of the enzymes. And so I talked with Gary Taylor, who had a PhD position available. Um, and that's how that started. So I ended up doing my PhD investigating through comparative structural analyses how hyperthermal stable metabolic enzymes manage to not fall apart at high temperatures and still maintain the chemical reactions that they have to do. Um, and so that was really interesting because the very highly thermostable proteins tend to be smaller, and they can often have higher oligomeric states than their mesophilic counterparts. And I guess this just really led me into a lot of wondering about how proteins regulate their stability, right? It seemed to me such a fundamental problem that whatever the environment, whatever the circumstance, and I hadn't really got any mammalian biology because this was all in archaea. But that's what led me to think about doing something to do with ubiquitination or regulation of protein stability in mammals. Biggest takeaway from that, I really loved my PhD. I would still say now, I got my PhD in 2001, so nearly 25 years ago. Um, I would still say that now it was the highlight for me. My supervisor just let me run with the project completely by myself. And I felt true ownership while still being guided. And it showed me how much fun a kind of scientific pursuit can be if you are truly allowed to be free to think and free to be creative and to come up with anything you want. And I'm not saying my PhD set the world alight, it didn't, but it was perfectly successful, taught me a lot of techniques, and really grounded in me a way of thinking physically about proteins.
Yeah. So, how was your PhD guide like? Like, so, what do you think as a human aspect of it, and as well as a scientist aspect of it?
It was really interesting because he was a physicist by training, which back in the day, a lot of crystallographers were physicists. They hadn't trained as biologists or biochemists. And so he he was also very busy. He was the head of a centre at the University of St Andrews. So your time with him was somewhat limited. Um, but it's like, I don't know how to explain. He trusted you, right? He just trusted. So I was too young to understand then what empowerment actually means rather than control. Um, but he empowered me. So he let me get on with doing what I was interested in. Um, as a human, he was a very gentle, say, not prone to offering criticism unless it was asked for. He was very happy to give criticism if you asked for it, but he wasn't somebody that would march around all day, every day, telling you what you were doing wrong. Um, and yeah, for me, it was a real, yeah, looking back and knowing some of the other experiences I had. I'm really lucky that that was my first proper experience of science, because it is how I think it should be. We should be following our curiosity, doing something hopefully useful, but mostly trying to solve a problem that matters to us, right?
Yeah, because that's what I also think. Because guidance is very important in your PhD life because it's early stages of your career. You don't know exactly what to do, how to do. So guidance and supervision becomes very, very critical in those aspects.
Yes, I agree to a large extent. I do think sometimes the hierarchy that you see in academic science leads people who are earlier on in their research careers to think that somebody else knows the answers. And actually, that's not research, right? If the answers are already known, that's just search, right?
Right.
So part of me thinks, yes, you need technical support or perhaps prompting to think about a question in a particular way. Um, but I personally, and I know lots of people do science differently than me, but I personally think it's a very creative pursuit, and that actually people coming up with crazy, wild ideas that they don't know whether they'll work or not is part of the fun.
Moving on from your post PhD career to your postdoctoral career. So you did your postdoctoral from St. Jude Children Research Hospital in US. So how is it different, like from your PhD research, like the culture and the environment like you did from UK and then you moved to US. So what
Yeah,
difference did you observe?
it was so different. Um, let's start with the culture. So bearing in mind, I grew up in different countries, and my father used to work for the British government, and so we moved around quite a lot. Um, there is a joke that when you are in America, even though you share the same language, you will have more in common with any other European than you will with an American as a British person. And I certainly found that to be true. I found the culture really quite alien. Um, the culture at work, St. Jude's Hospital was an amazing hospital, truly driven by a mission. And it was very, very well resourced. And that was great as a young scientist to be able to do any experiment I wanted to do. However, with that kind of resourcing often comes an expectation of almost factory-like productivity, I would say. Um, I had applied to work for a chair of a department who didn't have any positions available. But he said that there was a new PI starting and could he pass my CV on to her? And that PI was Brenda Schulman, who is now extremely famous. And I was her first postdoc. And I would say that it was a challenging time, both for her and for me, to be the founders of her lab and trying to get things started. Lots of lessons learned about how to make a lab out of nothing, and how to try to find projects that will be interesting enough but successful enough, and that balance between needing some sort of productivity and output, but also doing something important, which I still think is a huge challenge for any scientist. Um, I wouldn't say I spent 3 years and 9 months in America, in Memphis, and I do think if that had been my first experience of science, I might not have continued being a scientist. It was quite challenging. There is a very strong culture, not in every lab, but there's a very strong culture of presenteeism, that somehow it's about the hours you work and not the work you do in the hours you work. And I found that coercive and controlling and not a particularly enjoyable way to do science.
Working
It
in
was very
both.
successful though.
Yeah. So, as you work in both from a young, fresh scientist and an established scientist, so what do you think, as a student, when now if you go back, would you still choose, which one do you choose? So, for established scientist or, a fresh scientist who's be coming out of a postdoc?
Yeah, do you know, I don't know that it matters really. I think probably you should follow the question you're interested in. And I was interested in protein stability, so therefore Brenda's lab was a great lab to study that. Um, I think that some established researchers can be just as um, unaware of other people's needs, as earlier scientists can be. And I think that as I've gotten older, what I've realized is, particularly in science, because we have to interpret data all the time, I think that we as a group are quite judgmental. And I think that's probably where a lot of the problems in science come from, is that we should be judging data, but we shouldn't be judging people, right? And I think it's difficult for us to separate those two things. So I don't think it really matters if you work for a fresh. I don't think it really matters. So that one of the nice things about working for an established person is that usually it's a little bit easier to carve out your own thing to take with you because they're already well established. Working for a very junior person, they are usually quite frightened of, if you are, they need you to be successful, right? Because their success depends on your success. So they need you to be successful. And I think if it's done well, any any person who starts in your lab you should see them as your future peer, right? If they're going to do well, they should be a peer for you fairly
Absolutely.
soon. And I think you need to treat them like that from the beginning.
Thank you so much for this, because a lot of people tend to get confused, once they try to choose their PI, what to do, whether they should go for a young scientist or an professor or an established scientist. So this might be a very helpful insight. So that people should always focus on the science rather than on the scientist as well. So you also started your own lab in 2005 in Cancer Research Institute. So how was it? So, from being a student and then moving on to, mentoring them. So how was your experience? The first thought, like, I'm going to take one student. How was that? It's just,
Yeah. It was really scary. I don't know how else to describe it. I was very, very lucky because the package I got to start my lab up included two students and two postdocs. Um, so I had great resourcing to get started. What I would say about that is that there were all these weird rules that nobody told me about. So I tried to hire two students, and then somebody said, "Oh, you can't hire two students in the same year. That will be really difficult." And so suddenly, all that judgmentalism, everybody thinks that their way of doing things is the best way of doing it when it's not. It's just the best for them, right? Um, and then I was really fortunate because I got access to a coach who managed to help me understand myself. So I'm the oldest of nine kids, and there are only 15 years between me and the youngest. So there's a big group of kind of similarly aged people that I grew up with. And so for me, having two students start at the same time is perfectly fine. It's kind of my comfort zone, right? It's a normal thing to have people at the same developmental stage around each other. For somebody who maybe grew up with a older brother who was 5 years older than them, maybe they need a few years between PhD students, right? It's different for all of us. I found the establishing the projects quite hard as well. I worked at the bench for the first couple of years until I had my first child. And by that point, projects were up and running. And I think it took us quite a long time to publish anything, but I also think that the mid 2000s was a bit of a peak in terms of reviewers expecting three more papers worth of data whenever you submitted something for publication. I do think that has come down the other side since then, but I do remember being right at that peak when supplementary data was now a real thing. And suddenly it just seemed that no, because there's always another experiment you can think of, no paper was ever allowed to be finished. And I remember some of our work taking a very long time to publish. When I consider that the fundamental finding had not changed in 3 years, right? But more and more and more, bells and whistles were being asked to put on it. Um, it was a challenging place to work as well. One thing I would say for younger, if I had the chance to talk to my younger self, okay, like it was really great because it was a research institute with core funding, so I had lots of money to get started. But because it was Cancer Research UK, one of the projects I was working on, which I said I was going to work on when I interviewed, was on Parkin, which is not involved in cancer. Um, and then at every stage where I was reviewed, people would say, we need to get you to stop working on this. And I remember being very confused because I said I was going to work on it at interview and I was hired. So I took that to be tacit acceptance that this was what I would work on. Um, and it was a fundamental problem anyway. And I found that I realize now as I've gotten older that actually academic freedom doesn't really exist if you need somebody else to pay for it, right? It only really exists if you're paying for it
you are
yourself,
thinking,
right?
absolutely.
Yeah.
So, so from your experiences, I just one question just popped out in my head out of nowhere. It's just like how difficult it is for a young female scientist to come into science and then just adapt to the environment and understand that. So, how do you see in that aspect as well?
Yeah. How long do we have? It's this is difficult because I know some women who are older than I am who had to really struggle um, think it's they think it's not okay for women to talk about how hard it is for women because of the concern about putting other women off going into science. Um, Look, it's it's a difficult job, right? You have to constantly balance training new people, allowing them the freedom to be the scientists that they are, whilst also still holding on to the integrity of the scientist that you are. You have to convince people to give you money in a ever diminishing landscape. And the facts of the matter are that women are judged more harshly than men. Um, it's not just that. The default group is one particular type of man as well. And all other groups that don't fall into that default group are also judged more harshly than the default group is. Um, I think you just have to accept it as a reality. Um, I'll give you a silly example. That work on Park Inn that I was told to stop doing won me a relatively significant prize eventually, the Colworth Medal. And it's a medal from the British Biochemical Society, and I was the third woman in 52 years to be awarded it. And you would not believe how many people, not just men, how many people said to me, "Oh wow, they must have needed a woman this year." And I remember just sort of thinking in my analytical way, that interpretation doesn't make any sense. Because why wouldn't they have needed a woman last year, or the year before that, 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 or the year before that? It doesn't actually make any sense. The only thing that makes sense is that when a CV came along that was the best one, or whatever, And so I do not understand why we tend to think that when women do well, they've been the beneficiaries of positive discrimination. But when men do well, which they do disproportionately more often,
Absolutely.
that's
Absolutely.
somehow just merit based, right? So to me, as an actual scientist, I do not understand why these fallacies persist. What can I
Yeah,
say? I'm a very confident woman, and I think that confidence probably helps, right?
absolutely. The confidence, certain, people in the society tends to, overburden female scientists more than they will burden their peers along with that. So yeah, but people anyone in the science should have the confidence in them, and to pursue their ambitions, their goals, and that's the merit has to speak for itself. Instead of discriminating on any basis or any grounds, it's not fair to do it.
I just don't know how you undo the constant, pervasive discrimination that goes on all the time that people just refuse to accept happens. I think that's the problem. But anyway, I mean, I could talk all day about this. The overburdening of women, that's something I'm really discovering as I get older and more established, um, is how many times I am asked to sit on this panel, sit on that panel, review this paper, review this grant. And it's fine. I say no to at least three times as much as I say yes to. But it irritates me when, let's just take an example, a publishing house that is a for-profit publishing house writes to you, asks you to review something in 10 days, and you say, "I'm really sorry, I'm too busy." And then they say, "Can you suggest people from underrepresented communities? Since we, as a publishing house, are very keen to have full representation." And the paper you've just been asked to review is from six white blokes, right? So you as a woman have just been asked to review a paper from six white men, with the exhortation to please find yet another underrepresented person. And while I think it comes from a good place, of course it comes from a good place, right? Um, if only 20% of your workforce are female, and you're striving for 50-50 representation, you're asking 20% of your workforce to do 50% of the work. Everybody understands that that overburdens women. But what people don't consider is that you are also asking 80% of your workforce to only do 50% of the
Yes.
work. And what that then does is it frees them up to do the things like publish papers, write grants, etc., etc. And then we say at the end of that very, very unfair process that came from a good place, oh, what can we do? These CVs are better than these CVs. And I do feel that if it's okay to say we want the free service and the free labor from the underrepresented people, then if we trust those people's judgments on papers and grants and panels and all of the rest of it, then why don't we also say, okay, we're going to prioritize the work from those groups in our publishing house? But nobody will ever go to that point. So they'll take all the free stuff, all the hard work, and then shout, oh, discrimination, it has to be merit when it's actually when all of the studies show that the work from those groups is judged more harshly than the work from the default groups anyway. So I just feel like I don't want to sound really bleak, but until the default groups, whatever they happen to be, sometimes I'm in the default group as a white person, right? So, until the default groups truly recognize that they are discriminatory in their thinking, I don't think it will ever change. And I actually think you have to be a very confident, well-rounded, self-aware person to be aware of your own flaws and faults and bad things about yourself, right? Shall I stop now?
Let, let's let's move back to research. So, what currently you're working on ubiquitin, Parkinson disease. So, if you have to explain to a layman about your research, so can you please explain what's
I
working
will
on?
do my best. Um, so fundamentally, what I am interested in is a signaling system called ubiquitination, which exists certainly in all human cells, and it is the attachment of a small protein onto other proteins. And that attachment changes the chemistry and the physical nature of the protein it has just been attached to. Its most famous function is to alert the cell that the protein that has now been decorated with this other protein needs to be destroyed. And that happens when a cell needs to undergo cell division, for example, if you need some proteins to be switched on at this point, but switched on at that point in the cycle, tagging with ubiquitin is a quick way of destroying those proteins so that the next phase of the cycle can continue. However, it's also a signaling mechanism for so much more than just sending things to the bin. It's a mechanism for asking other proteins to come in to help with DNA repair. One of the systems we're interested in the Parkinson's field is most of the aggregated proteins that you find in neurodegenerative diseases are completely decorated with ubiquitin, and yet they haven't been destroyed by the bin, right? So for me, the fundamental questions I'm asking is how and when does a signal get created, right? So how does the cell know now's the right moment to put that signal on that protein? So to understand that, you have to understand how the enzymes that put the signal on interact with the proteins that are going to get the signal put on. You have to understand how close they have to be. You have to understand whether they need to be in different states. If there are other types of regulation that can influence that. And so for me, it's such a huge question. Some of the basics have been done now. We understand, as a postdoc, I worked on understanding how the ubiquitin itself gets activated so that it can be stuck onto other proteins. So we understand a lot about the enzymes that are involved. But I think in terms of understanding how those signals are created, we're still we're still in the dark ages, actually.
Let's moving on into your research. So, what do you think is the next, track or pathway for ubiquitin research, which you are working on? So, what's the future of?
My own personal research or where I think the whole field is heading. Um,
You can answer both of them. I would love to take.
I mean, I think, if I could wave a magic wand and really have a almost like a signaling points map that tells you when and where a given protein is going to be ubiquitinated, what the outcome of that ubiquitination is going to be. Um, I think that would be, I think that would just be amazing. If we could really see the whole signaling system as a complex thing itself. Obviously, as a structural biologist, I deal very much in binary, one-to-one events, trying to understand them, in their atomic, chemical detail. But I like to think that that is contributing to a signaling map eventually. Um, it's interesting seeing how the field is going because in I would say there've been two major things in the last 5 years or so, maybe 10. Um, I think the fundamental understanding of the system has reached a point where it's started to head into its application phase, um, which you see with the development of PROTACs, so proximity-based targeting chimeras, where a lot of companies are interested in exploiting the enzymes that stick ubiquitin onto target proteins to try to force an enzyme to be close to a target of interest rather than their cognate, natural partners. I think it's still, for me, quite early days as to how useful those PROTACs are likely to be. And I actually think that we probably still don't understand enough about whether proximity is really sufficient to drive degradation. And I say this particularly because, 95% of the enzymes that are used in these PROTAC developments it's the same enzyme, which makes me wonder just a little bit if we are accidentally following up something that is anomalous as opposed to generic, right? So I still think one of the major outstanding questions in ubiquitin biology is what makes a target a good target for ubiquitination, right? It would be so wonderful if we could predict ubiquitination sites, and we still can't. That being said, the other thing I think the field is really heading off into is really starting to appreciate the wondrous variety of ubiquitination events that can happen. So we now know you can get ubiquitin on sugars, that you can get ubiquitin on residues that are not nitrogen containing, right? So, serines and threonines, for example. There are reports of ubiquitination of nucleic acids. I mean, nobody knows functionally quite yet what is happening with those, but I think the fact that the chemistry allows for ligation to a reactive site, I think then actually the questions about, well, what makes a reactive site the best site to be targeted, become even more important. I worry a little bit that with the PROTACs, if you can drag something close to a protein that's got a lysine on the surface, yeah, sure, maybe it will get ubiquitinated and degraded. But wouldn't it be better if we could know that you use this enzyme if you want to target something that has a lysine in this kind of proximity, or this, not proximity, in this kind of shape or alignment, right? Wouldn't it be better to know both sides of the equation? So yeah, I think I also think still one of the major outstanding questions really is what in the neurodegenerative space is, what is that ubiquitin for? There were some really nice studies a few years ago, because I often thought, because of my protein stability background, that sticking ubiquitin on a protein gets thought of as a destabilizing thing because it leads to the degradation of that protein. But actually, ubiquitin is the most thermostable protein in humans. It has a melting temperature of 80 degrees.
Yeah.
Um, and actually, if you stick ubiquitin on something, you could be protecting that protein from unfolding. So it could be a stabilizing force as well as a destabilizing force. And there were some nice papers from the Friedman group a few years ago that suggested that that may be a mechanism. I'm not up to date enough to know if that's been followed up on, but I still think that's an interesting idea.
Yeah, I've read about and I've known, about ubiquitin a lot, and its TM, but never have thought in a direction where it can be used as a protective. That's brilliant. To move on in future to maybe as a use certainly in place of chaperone. Tried using it as chaperone, and then as well, and
It does, so it does have some use as a chaperone in terms of helping proteins to fold if you're trying to express them. And we use sumo for that, which is also very similar, for the same reasons, in fact. And I just think it's very interesting, the lid of the proteasome is covered in proteases, deubiquitinases. So the fact that it's removed rather than being threaded through the proteasome, I think speaks to its globular and very stable nature. Yeah, and I just can't help wondering if we sometimes the first function that is discovered is then set as the canonical function, when actually it's just the first one you saw, right?
yeah. A lot of opportunities in the field, especially in the with the evolving understanding of ubiquitin for us. So just out of nowhere to change a field. So a lot of people tend to believe that structure biology, structures, and as involvement of AlphaFold as well, and along with Rosetta. And so, what is your take on importance
Yeah.
of AI in the field?
All right. I'll say my provocative thing first, and then I'll tell you what I actually think. So, if I had a pound for every time somebody had told me that structural biologists are now out of a job, I would be able to run my own research institute, right? Um, it's not structural biologists that are out of a job, right? What AlphaFold has done, AlphaFold is incredible. Um, I would say I wish there was a little bit more recognition that it was only made possible by painstaking work of individual researchers working out experimentally the structures of proteins. Um, that being said, I think we are now in an era where most of the predictions AlphaFold make are not just reliable, they're brilliant. Right? And so for me as a structural biologist, it's been wonderful because suddenly things that I would have needed to experiment, I now don't need to do that. I would say the people who are really out of a job are the people who used to treat proteins in a structure agnostic way, where they would chop proteins up into, we just took 20 amino acids, then another 20 amino acids, then another 20 amino acids, did some pull downs, and therefore this bit is important for interaction, right? That cannot be done anymore. And I have to say that as a structural biologist, those sorts of studies I used to find them very challenging to understand because my brain would just immediately go, you don't know whether you've just disrupted the fold of that protein. You cannot come to that conclusion. So I would say that if anything, structural understanding is more important now than it ever was before, because no biologist now can ever think about any protein or system they're interested in without having to take into account the structure. And I do think that structural literacy is going to get much better. It needs to, but it's going to get much better. The role of AI, in science. Oh, I mean, because AlphaFold is so amazing, there's a bit of me that is super excited about the possibilities and potential. That being said, in my other role as an educator and someone who sets exam questions and marks questions for undergraduates, the generative AI, the text stuff, it's not very good. It's not showing evidence of thinking because, of course, it doesn't think. And it's, for me, quite shocking how quickly people are using generative AI without sort of realizing that it's not very good. I mean, it's brilliant in terms of working out probabilities of words being next to other words. But to give you an example, if I ask a generative AI to tell me about my research, it will quite happily suggest that I discovered ubiquitin, which I most emphatically did not. Um, and that is because every paper I've ever written has discovery of ubiquitin cited in there. So therefore, the probability of this being associated with me is very high, right? So I think in terms of revolutionizing, I don't know, uh, I still think there's no real substitute yet for someone thinking laterally about a problem. You said you thought the idea that ubiquitin could be stabilizing was a good idea, right? A machine's not going to make that connection because it won't look at thermostability and
Yeah. Yeah.
so I don't know. I think it's amazing, and we humans will figure out how to use it to our best advantage if we don't implode first.
Absolutely. Yeah. It's more technology is more about who uses it and how they uses it more than the existence and the known existence
Yeah,
of a technology and itself. So yeah, that's a very interesting take. So further, let's move on from this panel to another topic of discussions about so. As a scientist, when you take student, what is the primary thought, or the primary thing you think the student should have to pursue science? Or from your eye, how do
yeah.
distinguish from the student I want to take and this student, I can't.
Yeah.
So
So first of all, I wish that it actually was my decision. Um, Certainly in the UK system, PhD studentships tend to be managed at um, at least college levels, organizational levels. And because the world is competitive, we immediately start selecting people based on grades, which, as I've mentioned at the beginning, I did not get the grades to go to my university of first choice, although I still went there. And I don't know, I think one of the best scientists I ever worked with was a student who had come through a system in the UK, which was a university that used to not be a university. And there were other people involved in interviewing them who wouldn't have shortlisted them based on their background. Um, Whereas I read the cover letter. So for me, the cover letter is really important when somebody is writing to me. Um, I look for somebody who has some resilience. So I like to see what someone considers a challenge and how they consider moving on from that challenge. I always ask potential candidates, um, what does success look like for you? Tell me what your dream first year, dream second year, dream third year would be. Tell me what the highlight of your education has been so far, because I like to get an understanding. There's not a right answer, right? But I like to get an understanding. If someone says, "Oh, it was really hard for me to understand this technique," and then on the day I understood it, I was like, "Yeah, they're telling me something about their motivation and about what drives them." And people who say, "I was really pleased to publish this paper," they may be driven by finishing things or even by a bit of fame and glory, and that's okay as well. I need to know what motivates somebody. So for me, the non-successful ones tend to be the ones who are just very scholarly and want to tell me everything they know. Um, I'm much more interested in knowing what drives them. Um, that being said, I do still get it wrong sometimes with recruitment. Despite all the training I've had and all the effort, and I really do put a lot of effort into management, I still sometimes get it wrong. As in, I end up with somebody who somehow thinks that I'm a parent to them and I know all the answers to every question they will ever have. And I find those people very, very difficult to work with.
So, what advice would you give for upcoming PhD students or whoever is in PhD? Uh,
I think, when we talked about confidence, I think confidence comes from being authentic and believing in what you want to do and understanding that it's okay for not everybody to like that.